So our very first one is a cell viability assay. And what do we mean by cell-based assay? If you look, if you examine here, what is it that we uh, mean by a cell-based assay? That is,
Recording in progress.
what is it? Why isn't it called as an enzyme-based assay? Well, that's simply because a cell-based assay takes the intact cells from the tissue culture or from, let's say, the blood, and it uses those same cells to examine the performance, to examine the activity, to examine any activity that the cell would do while it is still living and viable. So that means it can be the cells which have a some kind of a span. They are still living. They are given the right living conditions. That means you are giving the cells, if they are mammalian cells, you are giving the cells 37 degrees centigrade. You never keep them in cold or too hot. You are giving them all the nutrients, etc. So in a cell viability assay, we are looking for these cells that are in tissue culture. Are they alive? Are they healthy and happy? Or if a drug, let's say a cancer treatment drug was given to them as a trial in tissue culture, then are they not happy? Are they dying? Or, so are they causing, is there any stress in them? So the result is that we're just trying to see if these mitochondrial enzymes are working or not. So the principle of this assay on the right hand side, if you take a look at it, we are simply looking to see is the cell essentially just doing its basic functions properly or not. So this is a cell drawing here on this side. Okay, the principle is showing it to you in a diagram. I think I have text later on. And the principle of this assay is you have cells that are in tissue culture, they are in medium, they're happy. Don't look on the left side right now. And these cells, they have a mitochondria. The mitochondria is the powerhouse. It generates ATP, it generates energy, it utilizes the oxygen and breaks down the glucose. And the mitochondria produces ATPs, and the cell gets all its energy from there. So MTT is a reagent, and you don't need to know the full name of this reagent. It's a chromogenic substrate, and this chromogenic substrate essentially is going to be added to the cells, and they are taken up. And you see how the membrane is like caving in. Well, this material simply goes and allows the MTT to go inside. And once it goes inside, the enzymes from the mitochondria, they convert this yellow color into purple. And the name of that chemical then is called as formazan. So formazan crystals are formed. And these formazan crystals actually then the cell can be broken down. And then when the cell is broken down, it releases this whole purple color. And here it is. Here is a 96 well plate. In this 96 well plate, you can see on the left hand side there are 96 wells, 12 on this side and 8 on this side. And more the purple color, the happier the cells are. There are many cells. That means there are many cells, and they are all their mitochondria is working fine, and they are all producing this purple color. And if you see light purple color here, that means there are less cells there, or they did not divide, and therefore they did not convert the color enough from yellow to purple. So that's kind of like the principle. And so all the same thing is just written in text here. Live cells with healthy mitochondrial function, they will perform the reduction of the MTT. They will reduce it to what? To formazan. And this takes all the kinds of enzymes, which we don't need to go into. It uses all of these enzymes. And that means those enzymes are working. If those enzymes are working, the cell is fairly viable. So it's called as a viability assay. And then anything that is changed to color, you put it into a machine, into a spectrophotometer, that measures the optical density at 570 nanometers wavelength. And so the more the color, the more the concentration of formazan. That means more the number of cells that were there in the sample. That means the cells were healthy and happy. And if the cells multiplied after 1 day, they're healthy and happy. So you can get the results. You can do this experiment and see how cells are surviving with a drug, without a drug. And so this is a full summary of how it is performed. And in this summary, let me walk you through this. In this summary, here's your, if you can see on the left-hand side is your, it's a tissue culture plate. So this tissue culture, it's not a plate, it's a flask actually. And at the bottom are all the cells, and that is liquid medium. And the cells in tissue culture are growing in this. So this is not your specimen from a human being. It is cells that we store in the freezer, and the cells are growing in here at the bottom. And so it's a flask, and this is how it's done, and it's laid down like this. So with using a aspirator, the cells are seeded onto this 96 well plate. So you see there's 12 holes, 12 wells on that side, and 8 wells on this side. And so small amounts, about, here it is, 2,000, 20,000 cells are seeded per every well. 2,000, actually, not 20,000, sorry, 2,000 here in this case, in each well. Because this is ml and it holds zero point 0.1 ml. And then the cells are grown in an incubator. This is an incubator at 37 degrees. It holds moisture. It has carbon dioxide that the cells need. And it is incubated for 48 hours. The cells are, let's say they're growing. You just want to see the growth of the cells. And then the cells are subjected. The cells are then um, treated. They're kept there for 48 hours, and then the treat cells are treated with a uh, drug, any anti-cancer drug, let's say. So you put in an anti-cancer drug in all of these at different concentrations, or whichever concentrations you want, and then they are again incubated at 37 degrees for another couple of days. And then all of that is removed, all the pink solution and is removed, and the cells are at the bottom. When the cells are at the bottom, then you add yellow colored MTT to all of them. So just with the cell multi-channel pipetter. And you add MTT to all of them, and then you incubate it for 4 hours, and then you remove it and you solubilize it. You solubilize it with a solvent, and they all just turn purple immediately. They turn purple, and all you have to do is put it in a plate reader, and you have to read the ODs, and you get the optical density, and you calculate the IC50 of the drug for this. And so, again, in totality, grow the cells, divide the cells into 96 well plate, grow them again for some time to keep them um, to get them normalized, and you do that in an incubator, and then you take them out and you add drug to each of them, how much drug you want, and again you incubate them for 2 to 4 days, and then you can 2 to 3 days, and then you can remove all the medium, but the cells are at the bottom. You cannot remove the cells are sitting at the bottom, and you add MTT to all of them, yellow colored MTT, and after 4 hours you add a solvent, and whichever is a happy cell, which is a growing cell, that will become more purple. That which is not growing, or it's dead because of the drug, therefore it will become less purple. And spectrophotometer reading will check the intensity of the color. So now I'm going to take you to this a YouTube video which kind of like shows you the same thing in a lab. Just thought you should be able to see it in a lab. I um okay, it's not working. Okay, let me show you this. Give me a second. So that's your 96 well plate. And in this 96 well plate, this person is somebody. Can you please turn your microphone off? Mute yourself, please. So cells have been added to this 96 well plate. This is a tissue culture hood. And you're growing the cells for 24 hours. One of the co-host, if you're the co-host, can you silence the one microphone of somebody which is on? So that is yellow colored MTT. So the cells have been growing or whatever it has been treated. This is for demo purposes. MTT is now added. The cells are sitting at the bottom of these wells. See these little bottom? They're just 0.1 ml volume, just 0.1 ml. And cells are sitting at the bottom and they are getting all the nutrients. And now MTT is being added to it. Abnova is a very good company. It sells a lot of products. Then put them in the incubator with moisture, etc. Yeah. So this is a multi-channel pipetter. So it can do 12 at one time. So this is about 100 microliters of that solvent dimethyl sulfoxide, anything that solubilizes it. And the cells immediately they break up and all that formazan crystals that we said, they all turn purple immediately. And those lanes that do not turn purple, that means they had less cells. And so that's your purple color for conversion of MTT by the mitochondrial enzyme. And this is a plate reader. It's a spectrophotometer. It will read it at 540 wavelength. And that way you get this. So I really like a lot of the Nova items. So I would like to just show you just for a bit how cell culture is done. Uh, let's say these are breast cancer cells or whichever. So just want to show you how cell culture is typically done. So these are from frozen cells. These are cells that have been just taken out of the liquid nitrogen, and you warm it, and immediately you have to put it in tissue culture medium. And so these are cells that are being taken out, and they are put into a tissue culture medium, and all of this is being done in the tissue culture hood. And this is a centrifuge. All the cells are taken out by spinning them, and you reduce the volume from the centrifuge. And this is that flask that I showed you. It's called as a T flask. And the cells are plated very gently. See how gently they're plating the cells? And this is red color is more medium. Add more medium to the cells. This has all the nutrition. It has the all the amino acids. It has um, all the growth factors, all the signaling molecules. And so now they're all mixed up. Actually, this being washed. The cells are being washed. Oh, they're not washed. She's picking up all the other cells and she's adding the remaining in here. Can you see how gently it's being done? No scratching, no scraping. Just go to the center and drop it in. The cells are very sensitive and they could be in clumps, so they're being mixed up and down, but not vigorously and no bubbling. And then they are they are right now under the microscope. They're all round. They need some time. And so after about just putting them in, giving them 37 degrees. And so those are all the cells that are being grown. Cell culture is a fairly expensive deal. And all those cells that you just saw, those round cells, are now stuck at the bottom. They are polygonal in shape. And so this is now, treatment of the cells. So the cells are all stuck here at the bottom. They're sticking at the bottom. So with that, I think just to show you how cells are treated. So again, she's putting in new medium. She's going to wash them and uh, put in maybe new medium, washing them, and she's going to separate the cells. So we don't need to see that part. So I will exit from here. And we will just go to what we did, what happens is that the OD that you get in here is just a measurement of the growth of the cells. And so that is cell viability. Cells are viable or they're not. Let's go to the next one. In the case of the next one, um, in the case of the next one, this is going to be a video. So this is a let's see if the cell invasion assay, and it includes some angiogenesis in it. And so remember we talked about cells being invading that basement membrane. So how can you do that? How do you test for those in a culture? So the way that I'm going to show to you is I'm going to show you a video, and it'll take you through that. So just for a second, what is the cell invasion assay? A cell invasion assay, it has to be done in the lab. You cannot examine this in tissues and remove tissues all the time. Oh, sorry. Okay. So you have to do this in the lab, and you have to find ways to replace the same thing in the lab. So in a cell invasion assay, we are looking for the cancer cell, to crawl, walk through the ECM, the extracellular matrix, and that has some collagen in it. So it will have, again, it will have your collagen fibers, or it simulates collagen fibers. And this is important for cancer metastasis, so that's why I included this. And so it is used in a transwell system. It's like a well in a well, a double-decker well. So I just showed you one well, and you make a double-decker, and I'll show you in a minute. And after incubation, the cells will by themselves, they will have crawled, they will have migrated, because cells can crawl, they can walk, they can crawl just that way that they like to do metastasis. So they migrate from the one chamber to the bottom chamber, from the top chamber to the bottom chamber. And that's all. So I think that's a very simple way of saying it. Let's look at the video and see.
Have
Let's
developed
look at the
3D
video.
models to more accurately study cell invasion and migration processes. While most traditional cell culture systems are 2D, cells in our tissues exist within a 3D network of molecules known as the...
Are you able to hear?
Scientists have developed 3D models to more accurately study cell invasion and migration processes. While most traditional cell culture systems are 2D, cells in our tissues exist within a 3D network of molecules known as the...
Don't know what happened. Let's see. Oh. So just to show you that what we do is we put the cells, I'm not sure why it's sticking there, but if we want to study this in 3D, so we put the gels at the in with a matrix, like a jelly substances. It simulates the collagen, it simulates the ECM. So that's where it is. It's stuck there for some reason.
exist within a 3D network of molecules known as the extracellular matrix, or ECM. While many of the mechanistic processes required for cell motility in 2D and 3D are similar, factors such as the reduced stiffness of ECM compared to plastic surfaces, the addition of a third dimension for migration, and the physical hindrance of moving through the mesh of long polymers in the ECM all present different challenges to the cell compared to two-dimensional migration. This video will briefly introduce the basic function and structure of the ECM, as well as the mechanisms by which cells modulate and migrate through it. Next, we'll discuss a general protocol used to study endothelial cell invasion. Finally, we will highlight several applications of 3D matrices to studying different biological questions. Let's begin by examining the composition of the ECM and how cells interact with it. The ECM performs many functions, such as providing support for cells, facilitating intercellular communication, and separating tissues. ECM composition varies among different tissues and has different biological properties, but it can be classified into two broad types. The basement membrane serves to anchor and separate tissues, while interstitial matrix surrounds and supports the cells within a tissue. The interstitial matrix is mostly composed of the fibrous protein collagen, but also includes elastin and fibronectin. Several biological processes need to occur for cells to migrate through the ECM. The first is cell matrix adhesion, which involves transmembrane proteins called integrins. These link the ECM to the cell's internal scaffold, known as the cytoskeleton. Another process is the structural rearrangement of the cell's cytoskeleton. This leads to the formation of specialized structures called invadopodia, which are protrusions of the cell into its surrounding matrix. The final step is ECM modulation. This typically involves degradative molecules known as matrix metalloproteases, or MMPs, which accumulate in the invadopodia and degrade the surrounding ECM, facilitating cell invasion. 3D matrix invasion assays allow scientists to visualize and study this complex process. Now that you're familiar with ECM and its interaction with cells, let's walk through a protocol for studying ECM invasion by endothelial cells to form tubules. By culturing endothelial cells in a 3D environment, one can simulate the biological process of blood vessel growth, also known as angiogenesis, which is important during both normal development as well as cancer. First, endothelial cells are cultured, and a single cell suspension is prepared by treating the cells with proteases such as trypsin, and passing them through a mesh filter to break up cell clumps. The 3D matrix, commonly composed of collagen, fibrin, laminin, or more complex combinations of these components, which can either be prepared in lab or ordered from commercial vendors, is then thawed on ice. Since most ECM preparations polymerize at higher temperatures, it's helpful to keep other equipment and reagents cold as well. The cell suspension is mixed with the thawed matrix solution to embed cells, and this mixture is placed into a cell culture incubator where the higher temperature will cause the matrix to polymerize. Once the cell-containing matrix is set, culture media containing angiogenic factors is added to the matrix dish. Using time-lapse microscopy software, individual cells can then be tracked to observe their migration through the matrix. The resulting images are analyzed and cell positions are used to calculate movement direction and distance in microns. These values can then be plotted to determine locomotory activity, the average migration rate of the cells. Finally, tube network formation is observed and analyzed using visualization software to identify features such as nodes, tubes, and loops. Now, let's explore a few applications of 3D matrices in specific experiments. Cell migration is mediated by active modulation of the cellular cytoskeleton. In this experiment, collagen matrices were prepared and mixed with a stain containing red fluorescent protein to allow for visualization. Individual cell spheroids, which are free-floating cell clusters, were isolated and embedded in the collagen matrix. Following incubation, the embedded cells were stained for specific cytoskeletal components and imaged by fluorescence microscopy. Researchers observed cytoskeletal components and their alterations as cells migrated through the ECM. Scientists can also study how the properties of the ECM affect migration. Using a concentric gel system where cells are embedded in an inner gel matrix surrounded by outer matrices of varying concentrations, scientists can track cells using time-lapse microscopy to study their migration from the inner gel to the initially cell-free outer gel. Researchers observed that the greater stiffness of higher concentration gels resulted in increases in both cell displacement and overall distance of cell migration. Finally, matrix invasion assays can be performed within a living animal to study angiogenesis in an organ-specific context. Here, fibrin gels, commonly used in tissue engineering due to their biodegradable nature, were generated, followed by implantation into mouse lungs, where the gels were held in place by a glue made of the protein fibrinogen. Cell migration and new blood vessel formation were allowed to occur for the following 7 to 30 days, after which the lungs and fibrin gels were harvested, fixed, and sectioned. Imaging of these sections revealed blood vessel and alveoli formation in the implanted gels, giving researchers insight into this crucial aspect of lung development in its in vivo setting. You've just watched Jove's video on extracellular matrix invasion assays. This video discussed the composition of the ECM and how cells migrate through it, presented a simple protocol to study endothelial cell migration through a 3D matrix, and highlighted several cellular processes currently being studied in the context of cell-ECM interactions. Because endogenous cell migration occurs in 3D space, these biological conditions are best simulated by 3D culture techniques. Improvements in matrix composition will continue to allow scientists to more accurately replicate and study cellular migration in the lab.
So what you saw there was, let's see.
As always.
So what you saw there was actually the transwell assays, probably in a similar but a different video. I didn't realize that the transwell is not there, but it was still the same concept. There were one well on top of the other was not there, but it's still the same concept. And the last one is a tunnel assay for apoptosis. So let's go through how apoptosis appears. I do want to show you just how the cells look when they apoptose. So let me just show you this one. This is going to be for you. Open it up.
Apoptosis, a form of programmed cell death, has been induced in these cultured cells. Cell death is characterized by blebbing of the plasma membrane and fragmentation of the nuclei. Suddenly, cells weaken attachment to the substratum that they have been growing on and shrivel up without lysing. In the following movies, we observe the process at higher magnification.
So in apoptosis, the cell commits a suicide, but it does not hurt anyone else. It's breaking up, nucleus broken. Completely. See, the nucleus will just completely break all the DNA material. There's membrane blebbing, and the cell just dies. Cell dies, but does not hurt the neighbor. So that's the beauty of apoptosis. Let me take you to the last video, which is the tunnel assay video. This is for cell death.
Tunnel assay is most commonly used to detect cells undergoing apoptosis, which is a form of programmed cell death. Apoptosis is an important biological process during development and for maintaining tissue homeostasis. Tunnel staining allows for visualization and quantification of apoptotic cells. This helps scientists test efficacy of new treatments for disorders in which apoptosis is either inhibited, like in cancer, or enhanced, as in neurodegeneration. This video will explain how the tunnel assay can be used to label cells undergoing apoptosis, a step-by-step protocol for performing this method in tissue sections, and how researchers are applying this technique to understand mechanisms of cell death. Before delving into the protocol of the tunnel assay, let's discuss the principles behind this technique. One of the many hallmark characteristics of apoptosis is DNA fragmentation. How does DNA fragmentation occur? Apoptosis is carried out by enzymes called caspases present in the cytosol. Their primary role is to cleave proteins in order to dismantle the cell. In addition, caspases activate an enzyme called caspase-activated DNase, or CAD, by detaching it from its inhibitor, ICAD. Activated CAD is an endonuclease that travels to the nucleus and cleaves chromosomal DNA. Cleavage of DNA ultimately causes accumulation of DNA fragments with nicked ends, and the tunnel assay fluorescently labels these nicked ends of fragmented DNA, allowing scientists to detect apoptosis. But how does this happen? For that, you have to understand the tunnel reaction. Tunnel stands for terminal deoxynucleotide transferase mediated DUTP ligand labeling. The two main tunnel reagents are terminal deoxynucleotidyltransferase, or TDT, and deoxyuridine triphosphate, or DUTP, which may be fluorescently labeled for ease of detection. In order to understand the tunnel reaction, let's go back to the apoptotic cells with DNA fragments. These niche fragments have free 3-hydroxyl groups. Once you add the tunnel reagents to a sample containing apoptotic cells, the fluorescently labeled DUTPs attach to these 3-hydroxyl groups with the help of the catalyst enzyme TDT. The cells stained using this procedure are called tunnel-positive cells, which can then be visualized using fluorescence microscopy. Now that you understand the basic principles and concepts behind the tunnel assay, let's outline a general protocol for performing this technique in tissue sections. The major steps of the tunnel assay include fixing the tissue of interest, permeabilization of the tissue, adding tunnel reagents, stopping the tunnel reaction, and finally, the analysis. First, the tissue of interest must be fixed in order to preserve biological structures. Fixation works by cross-linking proteins within cells. For the tunnel assay, tissues can be fixed by adding them to a solution containing 4% paraformaldehyde for 4 to 24 hours at 4 degrees Celsius. After fixation, cryosection the tissue into thin slices of 10 microns or less. The next step is permeabilization, which allows for reagents such as the TDT enzyme to penetrate the cell nucleus. Permeabilization of tissue sections can be performed by adding the tissue to proteinase K solution for 5 to 15 minutes at 37 degrees Celsius. Rinse tissue sections with phosphate-buffered saline on an orbital shaker for 15 to 30 minutes at room temperature. Following permeabilization, the TDT enzyme and fluorescently labeled DUTPs are added to the tissue sections along with a labeling buffer containing cobalt that acts as a cofactor for the tunnel reaction. Together, the tunnel reaction mixture and the tissue section are incubated for 1 to 3 hours at 37 degrees Celsius and protected from light to prevent the fluorescence from fading. Following incubation, stop buffer is added to the tissue section to cease the tunnel reaction, and after a short incubation, the sections are washed with phosphate-buffered saline. Finally, tissue sections stained using fluorescently tagged DUTP are visualized using fluorescence microscopy and assessed for localization of tunnel-positive cells within a given tissue. One can quantify cell death simply by counting the percentage of tunnel-positive cells in a given tissue section. Now that you've seen how to perform the tunnel assay to detect apoptotic cells, let's discuss how this assay can be used to address questions asked by cell biologists. Cell death occurs as a normal part of development for the sculpting of tissues and structures, and for the elimination of unnecessary cells. Therefore, scientists interested in this phenomenon study the effect of prenatal exposure to different substances on apoptosis during development. Here, scientists were interested in examining the effect of prenatal alcohol exposure on brain development. The results of tunnel staining performed on fetal brains revealed increased apoptosis in tissues that were prenatally exposed to alcohol, as compared to control animals. Scientists also used the tunnel assay to investigate apoptosis in response to bacterial infection. In this experiment, scientists developed a model of pneumonia by injecting mice with Pseudomonas aeruginosa, which induces lung inflammation. Then lung tissue was removed and tunnel staining was performed to examine apoptosis in response to the bacterial infection. Results show that apoptotic cell death increased in mice exposed to the bacteria as compared to control animals. Lastly, tunnel staining can be used on human tumor samples in order to determine tumor responsiveness to drugs. In this example, tumor samples were harvested from human patients and cultured ex vivo. Next, they were treated with preclinical drugs and assessed for a response using the tunnel assay. Data obtained show that treatment with a drug that inhibits heat shock protein 90 significantly increases apoptosis in tumor tissue.
So, someone has asked if you can get videos. So these are, I mean, these are kind of okay videos, and I think this whole program is being recorded, and so it is already here. So you will, if you want the recording for this talk, etc., so likely, it will likely be here. Okay, and otherwise you can contact me later on. I have to, these are all, there's legal binding to these. You have to purchase them, etc., etc. So I have to make sure that I follow all that protocol. Um, thank you for asking. It's at least I got somebody to chat a question. Alrighty. So, there was just a lot of information in there. I just wanted you to see the lab environment and things, how they work. Um, so this one just gives you an idea of, how cell apoptosis is visualized under the microscope, etc. So, most of these techniques, everything is kind of either based on fluorescence microscopy from tissue culture or simple staining so far. So I think that's where I would like to end. I do have some colony formation essay, but I don't know if that's going to make any sense. I just could not find a good way to show this. So I'm going to take this out, and um we will end it with these three essays. I'm not sure how much time I have used right now, but I want to thank you for being patient. And yes, that's right, Shorov. I think that they were already live streamed, but I don't know if there's a recording, so I don't know how that works. So if there is a recording, hopefully they will be there. But yeah, I have the permissions for these, and this is within the workshop, so no worries about that. So pretty much all good. And if any one of you need some of these, I can also give you some more information. The whole point was that you see some of the way that biologists do the work, and you see the cell staining, etc., things that we do, and then you think of things that could be useful. Anything that we do which is not tissue based, not human sample based, maybe those will be training grounds, for any kind of ML work that you do, any type of AI work, and that can be later on tested for something else. And I wanted to share this document that I created for you. So it's called as the additional materials. And I have here, let me make it big. I have here a little bit about the history. In my very first slide, I gave you a little bit about history of how it's named. And so there's this woman who's on TEDx talk who talks about that she uses paleoncology to understand more about cancer and, a little bit out of history. And I also went to zenodo.org where there is a collection, and I picked up some of the colorectal cancer stuff from there. Um, and it has about 5,000 images. So if you want to go in and you want to look at it, you want to use them, so please do so. I don't think I paid anything, or maybe I did, I can't remember. But it might need some permissions. And Kaggle.com is a private company, and in Kaggle.com I also picked up some of the colorectal slides early on yesterday. And here also, I think they give you codes, and there's a lot of things here. So this is typically for computer science folks. I think it'd be good. So give a Kaggle.com try and tell me, if me as a biologist I can use it. So let me know how you like that. Okay. And so those are the ones that I wanted to kind of share with you. And that's pretty much it for today. Um, so I would like to know your thoughts, and I'm happy to hear that